Hi everyone, we are the NeuroTDA team as a part of the Jahanikia Neurolab, and we are Krishnavani, Satya, Aparna, and Denise. So first, what is NeuroTDA? NeuroTDA is the application of topological data analysis, which is basically the shape of the data on neurodegenerative diseases and the genomics in order to help with drug discovery. We'll be talking about this further in our presentation. So first in this presentation, we are going to be first talking about genomes and specifically what genomes are. Secondly, we'll be talking about neurodegenerative diseases, so specifically what they are and the genes associated with them. Thirdly, we'll be talking about TDA or topological data analysis. So basically what is TDA and how it works. And then fourthly, we'll be talking about dimensionality reduction or Malper algorithm. So first on genomes. So let's review the definitions of basic knowledge. What is a genome? A genome is all the genetic information inside an organism. So genomes include DNA and RNA. DNA is a deoxyribonucleic acid, which is basically a two-stranded molecule that carries genetic information in humans. The RNA, on the other hand, is ribonucleic acid, and it is a nucleic acid present in all living things. Its primary function is to create proteins using translation and transcription, and it is similar to DNA, but it is only one strand. There are three types of RNA: mRNA, rRNA, and tRNA, which all have different functions. Then a gene is a segment of DNA that codes for a specific trait. What is genome sequencing? Genome sequencing is a laboratory procedure that determines the order or basis in the genome of an organism. Specifically, it's used to find changes in an area of the genome. Its advantage is that it can help treat and diagnose diseases. So the four steps of helping treating and diagnosing diseases. So the first step is DNA shearing, which is basically where the DNA is cut by molecular scissors. And then there is DNA barcoding, where scientists add small pieces of DNA tags to identify where each piece of DNA belongs. Thirdly, there's a whole genome sequencing, where the DNA is put into a sequencer that identifies nucleotide bases, which are specifically A, C, T, and G. And then the fourth step is data analysis, where the DNA is analyzed to identify patterns and compare sequences. Next, let's talk about gene expression. Gene expression is a process by which instructions in our DNA are converted into a functional product, such as proteins or other molecules. Some advantages is that it allows cells to respond based on its changing environment, and it also helps regulate the amount of proteins made. So there are two steps in gene expression: transcription and translation. So the first step is transcription, which basically uses RNA polymerase, which is basically an enzyme, to make an RNA copy of a gene's DNA sequence. Translation, on the other hand, is a second step, and it is a process by which the RNA is used to make proteins. The first step in translation is basically where the mRNA, or the messenger RNA, carries instructions to the ribosomes, and this message is then read by the tRNA, or the transfer RNA. The mRNA is read in codons, 3 at a time, which specify a particular amino acid. The tRNA delivers the amino acids to the ribosomes, which leads to the creation of a polypeptide chain, which then allows for protein formation. So towards the right, you can see a diagram of transcription and translation, which basically I've illustrated already, so we can just see the steps.
Okay, so now I'm going to talk to you guys about the neurodegenerative disorders that we're using in our data analysis and that we're going to apply the TDA and mapper algorithms for. Okay, first, Huntington's disease is an autosomal dominant disorder, which basically means that if a person has at least one allele of Huntington's affected, they have Huntington's disease. And so it causes the breakdown of nerve cells in the brain, and its symptoms include chorea, which is involuntary jerks or movements, dystonia, which is muscle rigidity or contracture, a difficulty talking and swallowing, impaired posture and gait, and a difficulty staying on top of things, lack of impulse control or awareness of behavior, and learning difficulties or slow processing. The genetics behind Huntington's disease include an inherited mutant CAG trinucleotide, the HTT gene on chromosome 4, which is shown in the picture on the right of the slide. And basically a mutant HTT gene, or MHTT, disrupts transcription and interferes with immune and mitochondrial function. The HTT gene has crucial roles in embryonic development, transcript regulation, and the formation of synapses. Next, we have Alzheimer's disease, which has more than 6.5 million patients in the United States. It's the most common type of dementia, and symptoms include memory loss, an inability to continue conversations or respond to the environment, getting lost in familiar areas, a difficulty completing familiar tasks, and misplacement of regular objects, as well as the repetition of activities. The gene that's mostly associated with Alzheimer's disease is the APOE4 gene. And 40 to 65% of patients with Alzheimer's have this gene, while 20 to 30% of all individuals have at least one copy of the gene, and 2% of all individuals have 2 copies of the gene. But it's not only the gene APOE4 that affects Alzheimer's disease. The buildup of beta amyloid, which is formed by the breakdown of the amyloid precursor protein, which then collects as plaques between neurons, causes the disruption of cell function and thus Alzheimer's disease as well. Next, we have Lewy body dementia, which is a progressive dementia that leads to a decline in mental ability and independent function. There are more than 1 million patients with Lewy body dementia in the US. Symptoms include visual hallucinations, memory loss, a short attention span, confusion, bradykinesia, which is slowed movement, insomnia and sleep disturbances, and emotional changes, including depression and anxiety. The gene behind Lewy body dementia would be SNCA, which codes for alpha-synuclein. And mutations in the gene lead to the formation of Lewy bodies, which are pretty much clusters of alpha-synuclein, and that leads to cell destruction. Parkinson's disease has about 1 million US patients and 10 million patients worldwide. Symptoms include tremors in the hand, arm, leg, jaw, and head, muscle contractions or stiffness, impaired posture, a lack of balance or coordination, bradykinesia, and speech or memory changes. There are multiple genes associated with Parkinson's, including LRRK2, PINK1, PARK2, PARK7, SNCA, and UCHL1. And basically, SNCA makes the Lewy bodies, and I talked to you guys about that in the last slide. And there are at least 30 mutations of SNCA that are linked to Parkinson's. And then PARK2 is a mutation of the gene that limits the accumulation of mutations in mitochondrial DNA. And that would cause an accumulation of damaged mitochondria, and that would lower ATP production, which is bad for the cells in the brain. Next, we have multiple sclerosis, or MS, and it's an autoimmune disorder where the myelin sheath or neuron coverings degrade, and the myelin sheath insulates the neurons, and thus MS causes some level of cognitive dysfunction. Symptoms of MS include numbness, tingling, vision problems, or an unsteady gait. And there are also multiple genes associated with MS. There's IL-7R alpha, IL-2R alpha, and FOXP3, CD33, and TMEM39A. IL-7R alpha and IL-2R alpha are both immune receptors involved in T cell action. FOXP3 and CD33 are polymorphisms associated with higher risk of multiple sclerosis, but there are more studies necessary in the field. And then TMEM39A has been found to have a statistically significant difference in expression for those with MS and those without MS. And the p-value for that is 0.0063, and usually anything below alpha equals 0.010 is statistically significant. There are also other neurodegenerative disorders like spinal muscular atrophy, familial fatal insomnia, and amyotrophic lateral sclerosis that I'm not going to go into detail on these slides, but we're also using data on those for our data analysis.
Hi, I'm Satya, and I'll be going through topological data analysis and just a brief intro. So what is TDA? And by the way, when I refer to TDA, I'm talking about topological data analysis, so you'll see that acronym a lot. But if you remember back to the goal of our project, it's to use
the presentation suddenly shifts. So I don't know, is it for me? Oh. So visually I can see like it has the shift a little bit to the right side. Perfect, good, thank you.
Yeah, so if you remember back to the goal of our project, it's to use the shape of our data and apply it to neurodegenerative diseases and genomics in order to find and aid in drug discovery, right? So our recurring theme here is going to be a lot about shapes. And so that's what TDA is all about. So it's a mathematical way to analyze the shape of our data sets. So this uses an algorithm called mapper, which is a bit computationally time consuming, but we will get to more on this later on the fourth section. So TDA is used in noisy data sets a lot. So this is high dimensional data, incomplete data such as biomedical data, where the shape takes a lot of dimensions and a lot of volume. And so metadata also accounts for this, which is basically data about the data. And for example, it's like the time and date of a picture. So if a picture, the data of a picture is like the colors and the pixels or the objects you can see, the metadata would be the time and date of that picture. And though the large volumes of metadata can take up a high volume of shapes, and that's why we use TDA to try and, discover and decompose that. So here's like a little example. As you can see, there's a bunch of data points here. The top one looks like a dinosaur. There's also a scatter of blobs. There's also a star. And so you can see that visually data can take a lot of shapes. And that's what we're focusing on here. We're trying to find the shape of that data so we can apply it to a field of neuroscience. So one of the first properties of NeuroTDA, and especially TDA in general, is called coordinate invariance. And this is a property that states that our data is independent of the coordinate system. So for example, a basketball is a basketball no matter where it is in the world, right? It still has that same shape. Similarly, the shape of our data will be the same no matter what position we put it in. So this means that we don't have to factor position into our calculations, and reference frames are unnecessary. So this is similar to how tensors sort of function in physics, where you don't need a reference frame to get that same answer. And so this means that the topological structure or shape of our data is retained no matter what. And this is important because we need to preserve that shape and save as much memory as possible, because all our data is very high dimensional and takes up a large volume of memory. So this is an example of coordinate invariance. As you can see, we have two images. Both of them represent the same set of points, just viewed from different angles. But as you can see, we can still see that circular shape of that data no matter where we put it on that coordinate point. And that's what we're trying to retain here. We're trying to retain that shape of our data, and that's going to be the recurring theme no matter what. So the second property is deformation invariance, and this basically says that topological features are unaffected by homeomorphism. And a mathematical definition of homeomorphism is that it's a continuous function that also has an inverse continuous function. And so taking the TDA analogy of this, it's sort of saying that the shape of a data set also has an altered or deformed version of the data set with a similar or traceable shape. And so, for example, what that means is adjusting a data set to stretching, deforming, altering doesn't change its shape or topological structure. So this means that we can use exploratory data analysis to cut and clean our data, but we can still retain that overall shape or at least keep track of it. And that is important because TDA uses lossy compression. And what that means is that since the volume of data is very high dimensional, we want to cut down that data. And by using lossy compression, we sort of lose some of that original data, but we can optimize how much we lose by retaining the same shape of that data. And since shape is all we care about in this project and the most important part of this project, that's what we want to be able to retain no matter what. And so this is a very complicated example of deformation invariance. So just to break it down a little bit, the top equation says F equals the little upside down triangle and an equation. So that refers to something called a gradient, which means you take the partial derivative of all the variables. And so this is basically a mathematical way to alter a function. And so as you can see under that little curvy arrow, you see that equation u equals x plus dx equals u of x plus du. So we're using something similar to u substitution here in order to change that original oval looking shape to something a little more deformed, but we can still keep track of that original oval shape because we know the mathematical steps we took to get to that deformed version. So by using the deformation invariance, we are keeping track of that original shape so we don't lose it in the process of using TDA. So the last and final property of TDA is compressed representations. And this is the ability of TDA to represent the noisy data in a compact way. So since mapper is computationally time consuming, we need to save as much memory as possible to counteract that. And so by using compressed representations, we can basically compact the data into something that will minimize the memory complexity. So that's what we'll get to on the next slide, a little image. So this is an example of a compressed representation. As you can see, the original solution space is just an array of like peaks and a bunch of colors. And with each level of encoding, we sort of narrow that down. But as you can see, we still keep that shape of those array of peaks. Those peak shapes are still there, and those colors are still there. They're just a little bit more compressed. but the original shape is still kept, and that's what's important because shape is the most important part to our project, and we need to be able to retain that.
Hi, I'm Denise, and now I will talk about some of the methods that we will be applying to analyze our data. The two main components of how we will implement TDA into our research are dimensionality reduction and TDA's algorithm mapper. So first, let's define dimensionality reduction. It's the process of reducing noise in high-dimensional data by decreasing the number of features or variables. Dimensionality reduction is really important for machine learning, data analysis, and predictive modeling. The data we use for these are often extremely large and high-dimensional, and unfortunately, noisy data can cause a lot of problems. Basically, data with an excess amount of noise can cause overfitting in machine learning, inaccurate data analysis, and an increase in time complexity. Luckily, however, dimensionality reduction provides a solution to all of these. There are many methods to reduce dimensionality in data, so I will only be covering a few of the most popular. The first is principal component analysis, or PCA. The way PCA works is it combines multiple variables into a single value through linear combinations, and this value is called a principal component. The algorithm creates multiple principal components, with the first being the most important. You can see on the graph to the right that they graph the first and second principal component against each other. This method is really helpful because it reduces dimensionality while still retaining the most useful information in the data. The second method is T-distributed stochastic neighbor embedding, or TSNE. The way that TSNE works is it models each high-dimensional object by a 2- or 3-dimensional point so that similar objects are closer together and dissimilar objects are farther apart. The last method I will be talking about is Uniform Manifold Approximation and Projection, or UMAP, which is the method that we will most likely be using to analyze our data. How UMAP works is it constructs a high-dimensional graph representation of the data and then optimizes it to a low-dimensional graph to be as structurally similar as possible. It has several advantages compared to TSNE and PCA. For example, it can cope with nonlinear scaling between variables unlike PCA, and it is also more efficient and can preserve more global structure. If you look at the graph to the right, you can evaluate the efficacy of UMAP compared to TSNE, and you should be able to tell how UMAP does a better job of clustering different groups together. And also, the fact that we can even evaluate how well the algorithm is working just through our vision goes to show how visual representations of data can be really helpful because of how easy it is to identify patterns visually. Next, I will be talking about TDA's mapper algorithm. Let's start with the definition again. Mapper is a function that builds upon any dimensionality reduction algorithm and produces a low-dimensional simplicial complex to represent the data. And you can think of a simplicial complex as just a set of points. Mapper is really useful because it can be utilized to identify interesting topological structures, and it can also provide insights to patterns in the data. The picture I have here shows an example of how Mapper can be implemented. Finally, I will be talking about how the mapper algorithm works. I've simplified the process into 3 steps, the first being covering the data with overlapping intervals, as you can see in the picture. Next, all points within an interval are collapsed into a single node. Finally, we connect the nodes that have overlapping intervals with an edge, which creates the final simplicial complex. You can see that the final product preserves the global structure of the data, although it does leave out some information, for example, that triangle in the middle right there. But that is all for dimensionality reduction and mapper. Next, we will be talking about some of the goals for our research.
So as Ms. Shanikia said, we're a relatively newer group. We were established in the summer of 2022, which is this term. And so we have some established goals. First, we want to apply TDA to sample data sets and eventually our own set to understand the shape and nuances of the data. And we've actually started on that goal this week. We're pretty much doing that right now. We also want to write and publish a paper about our algorithm and its results. And we want to present our research at 10 conferences and look for ways to further it. Thank you so much for listening. And on the right, we have a barcode for our citations. And if you have any questions, now would be the time to ask them.
Well done. Bravo. Good job. So fun fact, when I was at Stanford, my PI got R01 and K90, the top 2 grant over 1.5 million for implementing topological data analysis on MRI brain data. And I knew at some point, working on also as a part of a project, working on implementing TDA on FNIR's data. I knew at some point I want to come back to this topic, though it's pretty heavy, but I couldn't stop thinking about, using TDA. And because it was overlapping with my previous work, it took me a while to understand how I can find the application. One more fun fact, he's hiring postdocs to run TDA on his data in his lab. And now my high school students are trying to predict a subgroup of neurodegenerative disease gene expression using TDA that helps with drug discovery. So I am so proud of each and every one of you. It's a very, very heavy project. And I wish you best of luck. And the reason that you are here within like 6-7 weeks is because of your nonstop hard work in this project. So well done, bravo. So if you have any question for them, please go ahead. But don't ask about future works because we still have tons of work to do. Okay, next group. Thank you. Pop, 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 pop. Who are presenting? Beautiful. Okay, go ahead. The next one is all about creativity.
All right, hello, my name is Nicole.
My name is Pranjal.
And I'm Rashab.
And we are the Stick Tasks group. So first, we will be going into the background of our project, essentially what is creativity and what is functional fixedness. And then we will be talking about how this background knowledge applies to our project specifically. And then after that, we'll be talking about our creativity tasks and the ones that we are using. And we will have a little short game time to give you guys an idea of what our creativity tasks would look like. And finally, we would be talking about our next steps and goals for the end of this semester and the next semester. Okay, so I'm assuming most of you know what creativity is, but I will give a simple definition. So creativity is the ability to produce novel and original work. So I'm going to be talking about two main components of creativity, which is divergent thinking and convergent thinking. So divergent thinking is thinking that creates multiple solutions to a problem. So you basically have a problem and you think of as many solutions as you can. And convergent thinking is the opposite. It's basically thinking that narrows one best solution based on existing knowledge or a certain scope. So it's kind of similar to a multiple choice question. To give you guys a better idea of what divergent thinking and convergent thinking is, we came up with an example. So here you have a set of Legos. The problem is to create something, anything really. So a convergent thinker would use a manual as a reference to create a structure. For example, this Roman Colosseum. And divergent thinkers, on the other hand, just use their imagination to create anything. For example, they can create this dragon or this horseman or both if you have the ability to. And as you can see, neither is better than the other. Both divergent thinking and convergent thinking, can you go back? Sorry. Both divergent thinking and convergent thinking create beautiful structures. So what do? So our group thinks the most efficient way is to combine both, and you use divergent thinking to brainstorm ideas, and you find a manual similar to what you want to build, and you use that as a reference to build your own structure. Now I will be talking about functional fixedness. Functional fixedness is the main focus of our project. So this is really important. So functional fixedness is a cognitive bias that prevents the realization of new ways to use an object. So essentially, it's when a person is unable to think of another use of an object other than its typical use. For example, a pen is a writing utensil, and a person with functional fixedness would only think of a pen as a writing utensil instead of something else. And this definitely limits divergent thinking because it prevents us from thinking outside of the box. And we have another example. So here you have a leftover plastic bottle cap, and a person with functional fixedness would think that this is trash or recycle. However, there are many alternative uses to this bottle cap. For example, a person who practices divergent thinking and avoids functional fixedness would view this plastic cap as either a coin for coin flipping, this googly eye for DIY crafts, or even a tiny candle. And this is only one of the multiple potential solutions. Okay, so how is all of that information apply to our project specifically? So our project aims to understand how functional fixedness affects human creativity, and we will be doing that by administering multiple types of original creativity tasks based on tasks like AUT and RAT. And I'm assuming most of you don't know what that is, so now my teammate Pranjal will be going more into that.
Hi, so I'm going to go over various creativity tasks that we studied before we started making our own. So the first one is the Consensual Achievement Questionnaire, and it assesses achievement across 10 domains of creativity, and the level of achievement spans from no achievement in a particular domain to awards received or works published. So for example, maybe a certain person in music, they have like performed at a really prestigious symphony, then they would gain a lot of points for that. But maybe when it comes to writing, they haven't really accomplished much, outside of the basics. So then they wouldn't receive as many points. So according to your achievement in each of those domains of creativity, you gain more points. And this test is self-reported, and it's very easy to administer or score. The next test is the Inventory of Creative Activities and Achievements. And it's very similar to the CAQ, but the main difference is it assesses creativity across 8 domains instead of 10, and it is also self-reported. Next, we're going to be talking about the GO Personality Scale, which is a self-assessment based on personality characteristics. So this test contains 30 adjectives, for example, absent-minded, active, dominant, and each adjective is considered either positive, like this is a trait a creative person would have, or negative. The test taker checks off whichever adjective they feel like applies to themselves, and for each positive adjective, they gain a point, and for each negative adjective, they lose a point. So it's a pretty simple test. The next test is the Consensual Assessment Technique. So in this test, subjects are asked to create something. It might be a poem, it might be a short story, it might be some kind of small machine, and experts in the domain of whatever they're asked to create are then asked to evaluate the creativity of whatever product they make. So it's a little different from the rest of the test because you're evaluating an actual product rather than the steps taken to create that product. Next, we're going to talk about the Creative Solution Diagnosis Scale, which also looks at a product like the CAT assessment, but the main difference is that it doesn't require experts to judge it. It allows non-expert judges, so like potentially us. And it measures functional creativity. So an example of the CSDS might be asking participants to create a mousetrap. And the last test that I'm going to talk about today is the Divergent Associations task. So in this task, participants are asked to list words that have the least correlation with each other to score higher. So for example, if my words were like water and ice, I would score very poorly on the test because water and ice are very related to each other. They're the same thing at different temperatures. But if I listed water and a brick, I would score higher because those words are less related. But instead of 2 words, you list 10 usually. And this tests divergent thinking, as the name suggests, and it's also very convenient because it's a quick online test and it's been backed by a lot of research.
Then next we got the Alternates Use task, or the AUT, And essentially in this task, someone is given a everyday household object and they have to come up with as many alternate uses for that. And it's good for measuring functional fixedness. And one example of this was earlier in the presentation, we talked about the bottle cap, which someone would normally just throw it away, but someone with an open mind and without functional fixedness would be able to use that as maybe a coin for flipping or the base of a candle or other uses that someone could come up with. Next, we have the RAT test, and this is the remote associates test. And this is really good for measuring convergent thinking. And essentially in this task, what you're given is 3 words, and you have to look for one word that correlates between the 3 words. And there's also a visual RAT, which is similar to the regular RAT, except it measures more than just convergent thinking. And instead of having 3 words, you're given 3 images, and you're asked to see what correlates between them. For example, if someone's given a bottle of water, like a river and the ocean, the correlating word would be water between the 3. Next, we have the TTCT, or the Torrance test of creative thinking. And this was made by Dr. E. Paul Torres in 1966. And in this, there are 2 forms, the verbal form and the figural form. And 2 parts of the figural form is 1, there is the incomplete figure test, and 2, there's the AUT, which is the alternate use task, which I've already mentioned. So next we have the IFT, or the incomplete figure test, which is part of the TTCT. And it was also developed by the same person, and it requires the person to look at an image and finish it. So essentially a few stick lines could be used to draw an entire image and depict something much bigger. One of our last tasks that we'll talk about today is the consequences test, which is also part of the TTCT, and it was developed by the Amplitudes Research Project at the University of Southern California. And in this, subjects are asked to come up with as many consequences possible for a certain scenario. One example of this, like a task, could be, what if everyone just lost the ability to read and write? What would happen to everything? And a task that we're talking about in today's presentation is the EICT, or the Emotional Intelligence and Creativity Test. And this one can be administered 3 ways, digitally, verbally, or in person. And it tests empathy and problem-solving skills. And using a few pre-created scenarios, it lets individuals come up with some of their solutions.
Now to assess the pros and cons of each test, first we'll start with the CAQ and ICAA. These are the tests where you are measuring achievement in different domains. So the pro of these tests is that they're very commonly used, they're backed by a lot of research, and they're very easy to administer and score. But the con is that because they're measuring achievement rather than a specific process, people from lower socioeconomic backgrounds or people who've had less opportunities might be slightly disadvantaged. For example, it's possible that someone is like a great musician, super talented, but perhaps they never had the time or the money or the resources to perform in front of a huge audience. They might score lower than someone who's actually less creative. The next test is the Go personality scale. This was the one where participants checkmark characteristics that they feel apply to themselves and get a score according to that. Again, this is probably the easiest to administer and score out of all the tests, but we were concerned that it may contain some bias because I might feel that I am like very intelligent or very social, but other people might feel differently. So sometimes our perceptions of ourselves aren't 100% accurate. The next 2 tests are the CAT and CSDS. These were the ones where you measure the creativity of a product, like a poem or a picture. And the good thing about these tests is it's a very unique way to assess creativity, and it's one of those tests where experts tend to agree on what is more creative, what is less creative. But the con of these tests is that for the CAT, we need a panel of expert judges, which might not be easy for us to get. It might be a little inconvenient. And although we don't need expert judges for the CSDS, that does mean that the CSDS is like not quite as reliable as the CAT or some of the other tests we're going to discuss today. Then the divergent associations test. This is the test where you have to list words that are least correlated with one another. So again, this is a very quick, easy online test, which is a good thing. But the con is that it's kind of narrow and that it only measures verbal creativity. So it might be a good test when paired with other ones, but not on its own.
Next, we have the AUT, or the alternate use task. And then this task, one thing that's really good about it is it measures and explores functional fixedness. However, one of the biggest problems with this task is since there's no scoring guideline. One version of this task is actually used in the TTCT. However, in that there's a specified scoring guideline and that requires expert judges. So for the one we're later doing in this presentation, we have created our own scoring system, and that'll be mentioned later. Next, we have the RAT test, which measures convergent thinking, and it's pretty good at doing that. However, it only measures that. As such, it can't be used more than just a simple test. Third, we have the TTCT, which is one of the biggest tests used in measuring creativity. However, it's one of the longest tests, adding up to 90 minutes, meaning that someone who could be taking that test could be fatigued really easily. Next, we have the IFT, or the incomplete figure test, which is part of the TTCT. And in this, one of the biggest things is that it's an alternative to the IQ test. However, it has to be scored by expert judges, which makes it inconvenient for some people like us just doing our research on other people. Finally, we have the consequences test, which is simple to administer and it offers a lot more originality. However, for that, there is really no good way of scoring it, as someone might look at some consequences and some actions and be like, this is really good. However, other people might not think it's detailed enough. And we have the IECT, which is the emotional intelligence, and it measures that pretty well. However, it has to be created by us, and as such, it couldn't be as widely used.
Now we're going to actually play one of the games or take one of the tests that we talked about in our presentation, the AUT. So how this will work is when I reveal the object, you will have 1 minute to think of as many uses of the object as for the object shown and count how many uses you can come up with. We have two forbidden answers, weapon and decoration, because pretty much anything can be used as a weapon if you throw it at someone or as a decoration. So apart from those two uses and the common use of the object, anything is allowed. And once the time is up, tell us how many uses you came up with in the chat and feel free to tell us one of your ideas. Okay, your time starts now. So your object is an empty disposable water bottle with a cap. Okay, that's time. Please put the number of answers you were able to come up with in the chat. Ooh, I think the highest I've seen so far is 8. Very good.
No, that was 9. I would say pick up one of those who have 9 and ask them to list it. That will be exciting to see.
Oh, Jin has 11. That's a lot.
Okay, let's go with that one. Who's coming next?
Jin, if you would like, you can share the uses that you came up with.
Jen, can you come out of mute and share your answers with us?
There are, think about weightlifting, use it to watering the pads, to, putting the plant in like a container for putting the flowers, to growing fruits as well. And also use it to, for babies, like a rattle, and put paint in there as a seeing the turning with bees and water as a kind of a toy for kids. And I didn't write it down. So there was also the use it to like put hole at the bottom so that the water leak out as kind of a sprinkle wash the pads and stuff. And also put nutrition in there, add some holes so that nutrition slowly get out as a way to nutrient the plants. I lose count of how many I've done.
Those were some really good ideas. Thank you for sharing. I know we kind of put you on the spot, but I appreciate it.
Thank you so much. Yes, great. We have a lot of creative people on board too. I can see a lot of hate too. Good job.
Here are some of the ideas that we came up with. So it can be used as a tool to separate egg white from egg yolk. If you put it on like a plate and then you suck it up, it takes up the egg yellow. Like Jin said, if you poke holes in the cap, you can use it to water plants. You can grow plants in it, or you can even use it as a piggy bank. So as Rishabh mentioned, we came up with our own scoring guideline for the AUT. So how it'll work when we actually administer it is we will collect and list out all of the participants' answers, and then we'll identify any forbidden answers like weapon or decoration and cross those out. But we're also going to cross out any answers that are repeated by other participants. So for example, if I said, oh, it can be used to water plants, and someone else also said that, for both of us, those answers will be crossed out because we only want to look at unique answers that no one else came up with. Then your raw score will be the number of unique answers that remain after everything else is crossed out.
Okay, so next we're going to be covering the RAT test. And essentially in this test, as I mentioned earlier, you're given 3 words and you have to look for one word that's common between those 3 words. And it can go before or after the word. And for this test, what you're going to do is if you've come up with your answer, just type that answer in the chat, but don't send it. If you send it, everyone's going to see the answer and essentially ruin the test. So once I tell you guys the time is up, you guys can all send your answers in the chat and we'll try to see how many right answers people can get. All right, so the first test is sleeping, bean, and trash. Your time starts now. And essentially, you're looking for one word that's common between these 3 and without sending it in the chat. That's fine, I guess. I mean, I think that was the correct answer. I guess we can just skip this one then, since it's fine, don't worry about it. We have 2 more, it's all good, it's all good. So yeah, you did get the correct answer, so GG on that. Let's go with the next one. So that's main, sweeper, and light, and you guys have 30 seconds for this, and please don't send it in the chat.
And don't Google it too.
Okay, time's up. Whatever answers you guys got, put it in the chat. I see that street is the most common answer, and the answer was street, so you guys were all correct. Yeah. Okay, and the last one, time starts. Okay, and time is up. Whatever answer you got, put it in the chat. And most of you got that. Wow, that was actually a difficult one. Okay. Nice.
All right, thank you, Rishabh and Pranjal, for hosting that small game session. Hopefully you guys all enjoyed it. Now I will be going into the next steps for our project and what we hope to achieve by the end of this semester and next semester. So by the end of the semester, we hope to finish designing our experiment by creating more original creativity tasks and adding them to our current list. And we also wanted to mention that we did have more creativity tasks. We just didn't prefer to disclose them right now. And by the next semester, starting next semester, we hope to reach out to potential participants to participate in our tasks. And hopefully we can administer and hopefully begin our data collection process.
Thank you to Ms. Jahanikia for taking us in and guiding us throughout our new project. Thank you to all of our group mates who worked on this project with us and helped us with this presentation. And finally, thank you to ASDRP for giving us the opportunity to participate in this amazing research.
Well done, well done, well done. What an interactive presentation. Good job. To mention that our lab at the Society of SFNC, Society of Neuroscience of Creativity, we have our own lab. So that means that they're expecting us every year go and present some awesome, cool work to the Society of Neuroscientists that they work on the creativity. So we got our lab spot. So that is a great achievement for us. And we're going to have one after the other one, creativity. Of course, I should be able to sit and write a proposal for that. But something new and novel to share with the rest of Society of Creativity. I'm very excited for the team. They are making a novel creativity assessment, or I would say task. Again, we cannot disclose much information and details about it, but let's just wish them best of luck. And the expectation is way too high on this team. Okay, if you have any question, put it in chat for them. Thank you, team. Love it. And I'm sure everyone enjoyed the game time.